A very hearty welcome to everyone present for this session. So this is actually my first session, and since I am entering my PhD, I also wanted to start this series of delivering lectures for NEET UG because in recent years I have seen that there are a lot of inconveniences for people who cannot really devote the time or the money for expensive coaching classes. So I wanted to start this series so as to help people uh, from all the dimensions so that they can have a fair chance at clearing this medical entrance examination. So as for myself, my name is Ojasvi Singh and I'm currently a MSc PhD student at UNESCO DBT Regional Center for Biotechnology, which is located in Faridabad. I have cleared GATE BT in this year with an All India rank of 29. I have also cleared GATE Excel. I have also cleared CSIR lecturership with an all India rank of 36, and I have cleared DBT bed JRF. So JRF is a position which allows you to do a PhD and it gives you funding throughout. All right, so let's start with our first chapter. So I thought of starting with cell, the unit of life, because it is one of the very basic chapters that will help you build your concepts about biology, and it is a very important chapter. You will always have to remember these basics no matter what you do. Like if you take up medical, and if you do not take up medical but you choose other science, other areas of life sciences, it's a very important chapter. So let us start with this one. All right, and then we can cover all of the topics weekly, and somehow I hope that we'll be able to cover the whole of NEET syllabus. So let's start. So to start with, let's focus on what is a cell. So as you know that whenever we are talking about physical systems, we say that atom is the basic unit of physical systems. Similarly, in case of life sciences or biological systems, the basic unit is a cell, right? So, however, when you go deep into it, you'll realize that even the cell is made up of different biomolecules, and those biomolecules are eventually made up of atoms, right? So, let us start with what is a cell. So, unicellular organisms, which is like single cellular organisms, are capable of independent existence, like they can exist independently. For example, bacteria or yeast, which is a fungi, and performing the essential functions of life. Essential functions means basically being able to sustain itself and reproduce. Anything less than a complete structure of cell does not ensure independent living. For example, when you're talking about a virus, so virus requires a host to live, right? So hence cell is the fundamental structural and functional unit of all living systems. Anton van Leeuwenhoek first saw and described a live cell. So this is a very important point. Just mark it while you will be studying this. That Leeuwenhoek first saw and described the live cell. Robert Brown later discovered the nucleus. This is another point. So uh, first saw and described. So first saw was Leeuwenhoek and first described the nucleus was Robert Brown. All right. Okay. The invention of the microscope and its improvement leading to the electron microscope revealed all the structural details of the cell. For example, when you are looking through a microscope, what you'll be able to see will be, cell. And if you go into different types of microscopy, like there is confocal, there is fluorescence, then you'll be able to see different subcellular organelles. For example, you'll be able to see the Golgi, right? So this is all via light microscopy. If you want to see the very fine details like how the ribosomes are arranged, or even finer details, then you'll have to go to electron microscopy. Now let's move to the cell theory. So what is the cell theory? In 1838, Schleiden, uh, who was a German botanist, examined a large number of plants and observed that all plants are composed of different kinds of cells which form the tissue of the plants. At about the same time, Theodor Schwann. So till now we have two names, Schleiden and Swan. A British zoologist studied different types of animal cells and reported that cells have a thin outer layer, which is today known as the plasma membrane. So Swan gave the idea of plasma membrane. He also concluded, based on his studies on plant tissues, that the presence of cell wall is a unique character of the plant cells. That means that animal cells do not have a cell wall, plant cells have a cell wall, right? On the basis of this, Schwann proposed a hypothesis that bodies of animals and plants are composed of cells and products of cells. Schritt and Swan together formulated the cell theory. So the first two contributors of the cell theory were Schleiden and Swan. This theory, however, did not explain as to how new cells were formed. Now into the picture comes Rudolf Virchow. So Virchow says that cells divided and new cells are formed from pre-existing cells, that is omnis cellula e cellula. He modified the hypothesis of Schleiden and Schwann to give the cell theory a final shape. Cell theory as we understand today is this: that all living organisms are composed of cells and are products of cells, and all cells arise from pre-existing cells, right? So the contribution of Virchow was cellula, omnis cellula, e cellula. Omnis cellula, e cellula, right? Okay, so let us first take an overview of the cell. So you have earlier observed cells in an onion peel and or human cheek cells under the microscope. If you have not, you can try it on your own. Just you know, ask for help from your biology teacher at your school, and you tell her that you want to look at the cells present in your cheeks. So there is a process in which you have to use a toothpick and pick some cells from the inside of your cheeks and put it on a slide, and you'll be able to visualize it, right? So let us recollect the structure. The onion cell wall, which is a typical plant cell, has a distinct cell wall as its outer boundary, and just within it is the cell membrane. The cells of the human cheek have an outer membrane as a delimiting structure of the cell. Inside each cell is a dense membrane-bound structure known as nucleus. This nucleus contains the chromosomes, which in turn contain the genetic material. So just give me a second. So from whatever we have read just now, we can say that if this is a plant cell, this is the cell, and this is the cell wall present inside the plant cell. In comparison, this is the animal cell, this is the plant cell, right? So inside the animal cell, there is a nucleus. Even in the plant cell, there is a nucleus. Inside the nucleus are distinct structures known as chromosomes, right? So, for example, humans have 23 pairs of chromosomes, right? So, this nucleus contains the chromosomes, which in turn contain the genetic material of. So, over here, it is chromosomes, and chromosomes contain genetic material, which is the DNA, right? Let's move ahead. Cells that have membrane-bound nuclei are called eukaryotic cells. What does this mean? There are basically two types of cells. One is prokaryotic and one is eukaryotic. So in prokaryotic cells, you will find that none of their organelles have a double membrane structure. For example, the genetic material in prokaryotes, if this is the cell, the genetic material is dispersed in the cytosol or cytoplasm. Whereas in eukaryotes, the DNA is specifically enclosed within a double membrane structure, which is the nucleus. So the basic difference between a prokaryotic and a eukaryotic cell is that the eukaryotic cell consists of double membrane structures. For example, the nucleus or the endoplasmic reticulum, the Golgi apparatus, right? However, the prokaryotes do not. Okay. In both prokaryotic and eukaryotic, a semi-fluid matrix called cytoplasm occupies the volume of the cell. The cytoplasm is the main arena of cellular activities in both the plant and animal cell. Various chemical reactions occur in it to keep the cell in the living state. Besides the nucleus, the eukaryotic cells have other membrane-bound structures known as organelles. So, for example, the organelle that I was talking about, which is the Golgi apparatus or the endoplasmic reticulum, these are all membrane-bound distinct structures, and these are known as organelles, right? Lysosomes, mitochondria, microbodies, vacuoles. The prokaryotic cells lack such membrane-bound organelles. Now, this is a very important point that you have to remember, that the prokaryotic cells lack these membrane-bound organelles. Now, let's move on to what is a ribosome. So, ribosomes are non-membrane bound organelles. When we are talking about lysosome, when we are talking about Golgi, when we are talking about ER, right? Over here, we talk about membranes. When we are talking about ribosomes, ribosomes do not have a membrane. First, let me just rub off whatever I have written till now to make space for more information. You can take screenshots while I'm writing so that you'll be able to, capture everything that I've written. So ribosomes are non-membrane bound organelles found in all cells. They are non-membrane bound. It's a very, very, very important statement, right? So always remember that ribosomes do not have a membrane. And they are found in both the eukaryotic and prokaryotic cells. Obviously, prokaryotic cells do not have membrane-bound organelles, and since ribosome is not a membrane-bound organelle, it is present in the prokaryotes. Okay. Within the cell, ribosomes are found not only in the cytoplasm but also within two organelles, that is chloroplast and mitochondria. So, cell has ribosomes. Chloroplast, which is present inside the cell, also has ribosomes, and mitochondria, which is present inside the cell, also has ribosomes. Now, why do these two organelles also have ribosomes? We will come to it in further slides. All right. So, animal cells contain another non-membrane bound organelle called centriole. So, in the animal cells, in animals, there are two organelles which do not have membrane. One of them is ribosome and the other one is centriole, right? In plants, only ribosome is present and centriole is absent in the plants, all right? So, centriole, which helps in cell division, it gives a polarity to the cell so that cells can divide easily. Now, cells differ greatly in size, shape, and activities. For example, mycoplasma is the smallest cell, which is only 0.3 micrometer in length, while bacteria could be 3 to 5 micrometer. The largest isolated cell, blah blah. So these are some important points. However, the important points that you have to remember from this particular slide is, first of all, the cell theory, the contribution of each of the three scientists to the cell theory, right? So I'll just write it down. The first and most important thing you have to remember is the cell theory. And it is for your basics only, because a lot of questions do not come from cell theory in NEET UG, but you must know for the simple basic understanding of biology. It's a very important theory. After cell theory, you have to know which organelles are membrane bound, because a lot of questions come from this part, membrane bound organelles. Then you have to remember which organelles are present in prokaryotes. Then you have to remember about ribosome, that ribosome is not bound by any membrane, and centriole. So these are some of the important things you have to remember from this particular slide. Moving ahead, yeah, so I was saying that the largest isolated single cell of the egg of is the egg of an ostrich. Among multicellular organisms, human red blood cells are about 7 micrometer in diameter. Nerve cells are some of the longest cells. So till now, questions do not come from this part, so you can skip this part. But it is good to know for your own general curiosity. However, this part is not really important from the point of view of NEET UG. Now let's talk about prokaryotic cells. Now this is an important part, and a lot of questions come not only in NEET but also in other entrances like CUET and different other entrance tests for different other colleges. So prokaryotic cells are represented by bacteria, blue-green algae, mycoplasma, and PPLO, that is pleuropneumonia-like organisms. They are generally smaller and multiply more rapidly than eukaryotic cells. They may, okay, so over here I can give you an example. For example, in general, we consider E. coli for most of our experiments. Since I'm doing PhD, I will be using certain examples from there. So for example, E. coli, which is a bacteria, we say that it takes around 16 to 20 minutes to divide, depending upon the conditions it gets. Now, when you're talking about a human cell, it takes around 24 hours for a human cell to divide. Now see how many times faster the bacteria is multiplying compared to human cell. And it's because the prokaryotic cells are comparatively very less complex when compared to the eukaryotic cell. Okay. The four basic shapes of bacteria are bacillus, which is rod-like, coccus, spherical, vibrio, comma-shaped, and spirilla. This is an important part, but a lot of questions do not come from this part. So it's good to know for your own sake of curiosity and for your own understanding. The organization of prokaryotic cell is fundamentally similar, even though prokaryotes exhibit a wide variety of shapes and functions. All prokaryotes have a cell wall. Okay, now from here starts the important point. All prokaryotes have a cell wall surrounding the cell membrane except in mycoplasma. First important point. The fluid matrix filling the cell is the cytoplasm. There is no well-defined nucleus. The genetic material is basically naked, not enveloped. So over here, third important point, not enveloped by the nuclear membrane. In addition to the genomic DNA, many bacteria have small circular DNA. Fourth important point. These smaller DNA are known as plasmids. The plasmid DNA confers certain unique phenotypic characters to such bacteria. One such character is resistance to antibiotics. In higher classes, you will learn that this plasmid is used to monitor bacterial transformation with foreign DNA. Nuclear membrane is found in eukaryotes. Okay. Is found in eukaryotes. No organelles like the ones in eukaryotes are found. So this is the fifth important point. No organelles like the ones in eukaryotes are found in prokaryote cells except for ribosome, because ribosome is the only organelle which is not surrounded by a membrane, right? Eukaryotes have membrane bounded organelles. Prokaryotes have membrane less organelles. That means the organelles that are present in prokaryotes do not have a membrane surrounding them. So the only organelle that is present in both centrioles, the only organism that is the only organelle that is present in plant, animals as well as bacteria which is not bounded by any membrane is the centriole. Okay. So over here again I would like to remind you to read about centrioles. Alright. So prokaryotes have something unique in the form of inclusion bodies. This is the sixth point, which is inclusion bodies. Have something inclusions. A specialized differentiated form of cell membrane called mesosome is the characteristic of prokaryotes. This is the seventh important point, which is mesosome. Let me just write it. Mesosome. So what is a mesosome? It is a part of the cell membrane. So the cell membrane, it folds inside the cell membrane. If this is a cell membrane, so it folds inside and then eventually it is pinched off. So it forms the mesosomes. Characteristic, they're essentially infoldings of the cell membrane, right? Okay, so over from this point, this paragraph alone, you're getting 7 points, and questions have come from these 7 points. So these are very, very, very important points. So you have to make a note of all of it, right? Okay. So typical eukaryotic cell is 10 to 20 micrometer, bacteria cell is 1 to 2 micrometer, PPLO, and viruses. Now let's move ahead to cell envelope and its modifications. Most prokaryotes, especially the bacteria, have a chemically complex cell envelope. The cell envelope consists of tightly bound three-layered structure. Tightly bound three-layered structure. The outermost glycocalyx. So the outermost is glycocalyx, followed by the cell wall, and then the plasma membrane. Plasma membrane. Although each layer of the envelope performs distinct function, they act together as a single protective unit. So this is found in your bacteria, right? And all bacteria have what? They all have a cell wall, right? Which is the second part of the three-layered structure. Three-layered structure. Bacteria can be classified into two groups on the basis of the differences in the cell envelopes and the manner in which they respond to the staining procedure. So bacteria can be divided into two groups. One is gram positive and the other one is gram negative. So what do I mean by gram positive and gram negative? So the manner in which they respond to the staining process developed by Gram. So the Gram was name of a scientist and he had developed a staining procedure. So the bacteria which can get stained using Gram staining are known as Gram positive. The bacteria which do not take up the stain are known as Gram negative. So glycocalyx differs in composition and thickness among different bacteria. The gram staining is also possible because of the difference in the thickness of glycocalyx. It could be loose sheath called the slime layer. So glycocalyx, I'll just remove whatever I had written before. I hope that you all had taken a screenshot or made a note of it somewhere, right? So I just need to make some space to write some more things. So when I'm talking about glycocalyx, it can be of different types. And what are those different types? Yeah, so it could be a loose sheath called slime layer, like it feels somewhat like your capsules that you eat as your medication. In some, while in others, it may be thick and tough called the capsule, right? The cell wall determines the shape of the cell and provides a strong structural support to prevent the bacterium from bursting or collapsing. The plasma membrane is semi-permeable in nature. So we have talked about glycocalyx, we have talked about cell wall, now comes the plasma membrane. So the plasma membrane is semi-permeable in nature and interacts with the outside world. This membrane is similar structurally to that of eukaryotes. A special membranous structure is known as the mesosomes, which are invaginations of your cell wall, right? Cell wall, and this is your mesosome, which is formed by the extension of plasma membrane into the cell. These extensions are in the form of vesicles, tubules, and lamellae. Let's move ahead. Yeah, so when we are talking about different invaginations in the prokaryotes, so why do mesosomes form? So mesosomes have certain functions. They help in respiration, they help in secretion process, they help to increase the surface area of the plasma membrane and to increase the enzymatic content. In some prokaryotes like cyanobacteria, there are other membranous extensions into the cytoplasm called chromatophores which contain pigments. Okay, so from this paragraph only the role is important. DNA replication, distribution to daughter cells, etc. Bacterial cells may be motile or non-motile. If motile, they have thin filamentous extensions from the cell wall called flagella. So this is a very important paragraph. A lot of questions have come from this particular paragraph. We will be discussing the questions that have come in the previous 10 years after the after the session, so it will be easier for you also to understand which portions to read more thoroughly for your NEET, right? So flagella helps in motility. Motility, that is movement. Bacteria show a range in the number and arrangement of flagella. Bacterial flagellum is composed of three parts, that is filament, hook, and basal body. So you can remember this as filament, hook, and basal body, FBH or FHB, right? Filament, hook, and basal body. The filament is the longest portion and extends from the cell surface to the outside. Besides flagella, pili and fimbria are also surface structures of the bacteria, but they do not play a role. Do not. Okay. So it's a very important word because a lot of questions have come from this part and students often get confused. So when we're talking about the pili, its main role is in reproduction of bacteria. And when we are talking about fimbri, its main role is in holding on, right? So fimbria bristles like fibers sprouting. Some bacteria are known to help attach. So fimbri is attachment. For example, if a bacteria is attached to a rock, it will be via fimbri, right? So pili and fimbri are not used for movement. They are used for reproduction and attachment. Now let's come to ribosomes and inclusion bodies. But first, let me just remove whatever I've written to make space for more notes. Yeah. So as I said, ribosomes, they do not have a membrane. So they are membraneless. And they are present both in eukaryotes as well as prokaryotes. Inclusion bodies are only present in prokaryotes. Right, so let's just read about it. In prokaryotes, the ribosomes are associated with the plasma membrane of the cell. So in prokaryotes, the ribosome is associated with the plasma membrane of the cell. They are about 15 nanometer by 20 nanometer in size and are made up of two subunits, which is 50S and 30S. So 50S plus 30S. This makes up a whole ribosome in case of a prokaryote. So, which are present together to form a 70S prokaryotic ribosome. Together they make 70S. Okay. Ribosomes are the site for protein synthesis. Several ribosomes may attach to a single mRNA. So, when there is a single mRNA and there are multiple ribosomes attached to it, right? This is how it looks. Now, this is known as a polyribosome or a polysome. So, when we are talking about the 50S and 30S, it looks somewhat like this. This is the 50S, this is the 30S. Together they form 70S, right? 50 and 30. And from this part passes a mRNA. And together this whole structure is responsible for producing proteins. Okay. So inclusion bodies, now that we have discussed about the ribosomes, I'll just quickly remove this and we'll move on to the inclusion bodies. Just give me 1 minute. Yeah. So what are inclusion bodies? Inclusion bodies are basically small spherical structures which store the reserve material in prokaryotic cells. So reserve material in prokaryotic cells are stored in the cytoplasm in the form of inclusion bodies. They are not bound by any membrane system and lie free in the cytoplasm. Till now, in prokaryotes, we have studied structures like ribosome, which does not have a membrane. Now we are studying inclusion bodies, which does not have a membrane. And the third thing is the DNA. The genetic material does not have a membrane bound structure. So they lie free in the cytoplasm. Example, phosphate granules, cyanophycean granules, glycogen granules. So questions have come from this part. The names of certain inclusion bodies, right? So mark this. It is important. It might come in your exam. That is your NEET UG or any sort of entrance test like ICAR tests, right? Gas vacuoles are found in blue, green, purple, and green photosynthetic bacteria. So gas vacuoles are also inclusion bodies, right? Now let's move to eukaryotic cells. Till now we have been discussing about prokaryotic cells. Let me just quickly rub off to make space. Yeah. So let's move to eukaryotic cell. The eukaryotes include all protists, plants, animals, and fungi. In eukaryotic cells, there is an extensive compartmentalization of cytoplasm. That means the whole cytoplasm is divided into compartments based on the functions that these different compartments serve. Right? So, eukaryotic cells possess an organized nucleus. Let me just draw alongside. If this is a eukaryotic cell, it has an organized nucleus with a nuclear envelope, right? With a nuclear envelope. In addition, eukaryotic cells have a variety of complex locomotory and cytoskeletal structures. Their genetic material is organized into chromosomes like this. All eukaryotic cells are not identical. Plants and animal cells are different as the former possesses cell wall. That is, plant cell has a cell wall. It has plastids and a large central vacuole. If I'm talking about a plant cell, it has a cell wall, right? This is a cell wall. It also has plastids like chloroplast and a large central vacuole. And this is, for example, the nucleus. This will be the vacuole. On the other hand, animal cells have centrioles which are absent in plant cells. So animal cells have centrioles which are absent in plant cells. Let us now look at the individual cell organelles to understand their structure and function. Just give me a second. All right, yeah. So let's move back to it. Now let's move to cell membrane. The detailed structure of the membrane was studied only after the advent of the electron microscope. So before electron microscope came into picture, people could see that there was a cell membrane, but they did not understand the detailed and delicate structures that are present on the cell membrane. So meanwhile, chemical studies on the membrane, especially in human red blood cells, enable the scientists to deduce the possible structure of plasma membrane. Before electron microscopes came into picture, RBCs were used for experiments to find out the nature of the plasma membrane. These studies showed that cell membrane is composed of lipids. So the outermost membrane in an animal cell, which is the cell membrane, is composed of majority lipids, which is oil-like structures that are arranged in a bilayer. So this is how a lipid looks like. It has a head and two tails. And as mentioned, it was found out that the cell membrane is a bilayer, right? So this is the first layer, this is the second layer. Together they make a bilayer. Okay. Also, the lipids are arranged within the membrane with the polar head towards the outside. So this is the polar head, the circular structure, polar head, which is outside. It is interacting with the outside environment towards the outer side, and the hydrophobic tails toward the inner part. And these two tail-like structures, these are the hydrophobic tails. Okay. This ensures that the nonpolar tail of the saturated hydrocarbons is protected from the aqueous environment. The lipid component of the membrane mainly consists of phosphoglycerides, right? So mostly these structures are what? They are phosphoglycerides, and these are lipids. Okay. Later biochemical investigation clearly revealed they also possess protein and carbohydrates. So the cell membranes also consist of proteins, obviously, and carbohydrates. Now I will just show you how, for example, the proteins are present. So if this is the first layer, this is the second layer. So sometimes between these layers there is a protein which allows for the movement of water or ions or any other thing. Sometimes proteins are present embedded in one of the layers with their some receptor sort of protein extending out, so that whenever there is an extracellular signal, it can come and activate this protein, which will further cause more signaling. So these are just two types of proteins that are found in the plasma membrane. There are many other types of proteins, and we can discuss those proteins in upcoming classes. If you feel it's necessary, please let me know. So the ratio of these proteins and carbohydrates and everything vary according to the cell type. Depending on the case of extraction, membrane proteins may be categorized as integral or peripheral, while the integral proteins are a part of the membrane. So this part is as such not that important for your NEET UG or competitive exams. However, you have to remember that the cell membrane is a bilayer. It consists of phosphoglycerides mostly, and the hydrophilic head extends to the extracellular sides, and the hydrophobic tails are present in between the layers, protected from the outside environment, right? So this was mostly it. Yeah, so as we have talked about the structure of the cell membrane and different proteins present on it, over here you can see the integral proteins. So for example, in this case, it looks like a protein which is responsible for either transport of water or transport of ions, uh, or since it also possesses these sugar moieties, it can also participate in, signaling. So it can be either a transporter or a receptor. When both of these things act together, it is called a transceptor. All right. This is the lipid bilayer. This is the hydrophilic head, and these are the hydrophobic tails. As you can see, all of these these double structures, these are your hydrophobic tails. This is your protein which is embedded in the cell membrane. This is the sugar moiety that is present on these proteins. These sugar moieties have various roles. It can either attach some sort of glycoprotein to it, or it can play a role in receptor like functions. And similarly, they have various other roles. We can look into it as and when required. For now, let us just focus on the part that will come in the examination for now. If you have any more questions, you can always let me know, and I will discuss those questions in details. So now let's move into cell wall. So as you may recall, a non-living rigid structure called a cell wall forms an outer covering for the plasma membrane in fungus and plants. Cell wall not only gives shape to the cell and protects the cell from mechanical damage and infection, it also helps in cell-to-cell interaction and provides barrier to undesirable macromolecules. For example, since your cell membrane is semi-permeable, it does not play a role as much as cell wall in preventing the entry of certain macromolecules. Algae have a cell wall made of cellulose, galactans, mannans, and minerals like calcium carbonate, while other plants, it consists of cellulose, hemicellulose, pectins, and proteins. The cell wall of a young plant, the primary wall is capable of growth, which gradually diminishes as the cell matures, and the secondary wall is formed on the inner side of the cell. Questions have not come from this part in NEET UG in the past 10 years, so we'll be skipping this. I mean, you can read it obviously for your own curiosity and for your own knowledge, but questions have not come from this part as much. So we'll be moving forward. The middle lamella is a layer mainly of calcium pectate, which holds or glues the different neighboring cells together. The cell wall and the middle lamella may be traversed by plasmodesmata, which connect the cytoplasm to neighboring cells. Over here, the only thing that you need to remember is when two cells are interacting with each other, there is a bridge-like structure formed between them, which is known as plasmodesmata. So you have to know the role of plasmodesmata. So plasmodesmata is what it is. It forms a connection between two cells when there's a presence of cell wall. Okay. Over here, this is the function. So this may be important. It has not really, I don't remember seeing a question like that for the past 10 years, but This is something that you can always remember because it is an important information. So an improved model of the structure of cell membrane was proposed by Singer and Nicholson, widely accepted as a fluid mosaic model. It is a very important model and you'll be studying about it in great details if you ever take up biotechnology for your future. According to this, the quasi-fluid nature of lipid enables lateral movement of proteins within the overall bilayer. That means lateral movement means what? If this is the membrane, so if a protein is over here, then the protein eventually comes here, then it may go back to its original space or it may move further away. So the movement on top is like a sea and there's a boat in it. So the movement of the boat, something like that. So that's the lateral movement. Okay, lateral movement of proteins within the overall bilayer. This ability to move within the membrane is measured as its fluidity. How fluid the membrane is. For example, it will be difficult to move a boat on a road compared to moving a boat in the, what can I say, in the sea. So sea is obviously more fluid than a road. So boat is able to move better in the sea. So that is what defines your fluidity. Uh, However, fluidity is a very deep topic and we'll study more about what makes a cell membrane fluid in the upcoming slides or in the upcoming classes whenever it seems necessary. So the fluid nature of the membrane is also important from the point of view of functions like cell growth, formation of intercellular junctions, secretion, endocytosis, and cell division. Obviously, the more fluid the cell wall is, it will impact its functions. A rigid cell wall will not allow the reproduction to be as fast as comparatively fluid cell wall would. So one of the most important functions of the plasma membrane is the transport of the molecules across it. The membrane is selectively permeable. A very important point, it is selectively permeable to some molecules present on either side of it. Many molecules can move briefly across the membrane without any requirement of energy, and this is called passive transport. Neutral solutes may move, this is important. Neutral solutes may move across the membrane by the process of simple diffusion along the concentration gradient, that is from higher concentration to lower. It is for neutral molecules. Water may also move across this membrane from higher to lower concentration. Movement of water by diffusion is called osmosis. So for water to move, it is known as osmosis. As the polar molecules cannot pass through the nonpolar bilayer, they require carrier protein of the membrane to function to facilitate their transport across the membrane. A few ions or molecules are transported across the membrane against their concentration gradient. Such a transport is known as active transport. So whenever we are talking about ions, it will follow active transport. When we are talking about water, it will go via osmosis. And when we are talking about neutral compounds, like for example, let's say any particular lipid, so all those things will go via passive transport, right? Now let's talk about endomembrane system. This is a very important topic. Questions have come from this topic almost every year, and I don't think they are going to stop. It is an important topic anyway. So while each of the membranous organelles has important structure and function, many of these, yeah. So I'll just repeat what I have talked about till now because the system got disconnected. All right. So the endomembrane system includes endoplasmic reticulum, Golgi complex, lysosome, and vacuoles. So as I said, this paragraph is the most important paragraph till now because it's talking about the endomembrane system, right? So the endomembrane system has only four parts. Remember it. The first part being the endoplasmic reticulum, the second part being Golgi, the third part being lysosome, and the fourth part being vacuoles. All right. The so electron microscopic studies of eukaryotic cells reveal the presence of a network of reticulum of tiny tubular structures scattered in the cytoplasm, that is the ER. Hence, the ER divides the intracellular space into two distinct compartments. One is the luminal and the extraluminal. The ER often shows ribosomes attached to their outer surface. The ER reticulum bearing ribosomes on the surface is called rough endoplasmic reticulum. In the absence of ribosomes, they appear to be smooth. So ER is of two types. One is rough endoplasmic reticulum and one is smooth endoplasmic reticulum. Rough endoplasmic reticulum has this is how it looks. So it has ribosomes attached to its outer side, right? On the outside, you will find ribosomes. So RER is frequently observed in the cells actively involved in protein synthesis, because obviously ribosome is involved in protein synthesis. So whenever you see RER, there will be more amount of ribosomes, so there will be more amount of protein synthesis in those cells. They are extensive and continuous with the outer membrane of the nucleus. The smooth endoplasmic reticulum is the major site of synthesis of lipids. So when we are talking about smooth endoplasmic reticulum, its major function is lipid synthesis. Okay. In animal cells, lipid-like steroidal hormones are synthesized in smooth endoplasmic reticulum. Lipid and hormones are synthesized in the smooth endoplasmic reticulum. And which type of hormones? Lipid-based hormones like steroid hormones. All right. Now let's talk about the Golgi apparatus. So Camilo-Golgi first observed dense stained reticular structures near the nucleus. They were later named Golgi bodies after Camilo-Golgi. They consist of many flat disc-shaped sacs or cisternae. These are stacked parallel to each other. Varied number of cisternae are present in Golgi complex. The Golgi cisternae are concentrically arranged near the nucleus with distinct convexes or the forming phase and concave trans or the maturing phase. So the maturing phase is known as trans and the uh, convex phase is known as the cis phase and trans phase. Alright. And then again Golgi is again a part of the endomembrane system. The major function of Golgi is adding sugars to your proteins. So over here, basically glycosylation process happens. Glycosylation, that is the function of Golgi. So this glycosylation helps in labeling where the protein will go eventually. Like it gives it. So what happens is the ribosome produces the proteins. So the ribosome is, for example, making the cake. Now the cake enters inside your ER, that is the endoplasmic reticulum, and some decorations are done on the cake in the form of certain modifications that happen inside the endoplasmic reticulum. Now that endoplasmic reticulum carefully keeps the cake inside the box and sends it to the Golgi. Now when the cake is received by the Golgi puts an address stamp on the cake parcel so that the delivery man can know where to deliver that cake. So that the address postage is called glycosylation. That means inside the cell on the proteins, some sugar moieties are added. That those sugar moieties help in identification of the target for the protein to go and attach to or get inside, right? So you'll study more about it in your upcoming classes or in your higher classes. Let's move ahead. So as I was talking about your plant cell and animal cell, let us quickly go through them once. So rough endoplasmic reticulum is present. In your plant cell, whereas centriole is present inside your animal cell. The centrioles help in proper polarization of the cell and helps in cell division. Peroxisome is also present. Lysosome is present. Ribosomes are present in your animal cell. Microvilli is present. For example, this cell can particularly be a cell of your, for example, small intestine. Small intestine. Right, and plasma membrane is present in both of them. Golgi apparatus is present in both of them. Nucleolus, nucleus is present in both of them. The differing parts, for example, over here are plasmodesmata, because plasmodesmata will not exist in animal cells, because plasmodesmata occurs whenever there is a presence of cell wall. Cell walls are present in your plant cells. All right. Okay, and moving ahead. Yeah, so this is basically it. Cell wall over here, then vacuole, a large central vacuole. Large and central vacuole is a part of your plant cell. Then again, okay, so this was mostly it. Chloroplast is also present in your plant cell only and not in your animal cell. Mitochondria, however, is present in both plant and animal cells. I would like to talk to you all about a thing called endosymbiont theory. So for now, I want all of you to go and find out what is the endosymbiont theory. I want you to realize which organelles are a part of this endosymbiont theory and why it exists. And if you have any questions, you can always come and ask. All right, moving ahead. So let us talk about the cis and trans phases of the Golgi. The cis and trans phases of the organelle are entirely different but interconnected. The Golgi apparatus principally performs the function of packaging materials to be delivered either to the intracellular targets or secreted outside the cell. Materials to be packaged in the form of vesicles from the ER fuse with the cis phase. So this is how it looks like. For example, this is the ER. This was your, nucleus. This is a double membrane structure of ER, right? So this was your nucleus. These were your DNA, hereditary material. This were your ribosome. So from the hereditary material, that is your DNA, comes the mRNA. This mRNA gets attached to one of the ribosomes and protein is synthesized. Once the protein is synthesized, that protein comes out of the ER and goes to the Golgi, right? It enters the Golgi. Inside the Golgi, it is further processed and processed and processed and finally packaged into vesicles. So this is the protein that is packaged into the vesicle. This whole thing forms a system, right? So this explains why the Golgi apparatus remains in close association with the ER. A number of proteins synthesized by ribosomes on the endoplasmic reticulum are modified in the systemy of the Golgi apparatus before they are released from the trans phase. So this is a trans phase. uh, Golgi apparatus is the important site for formation of glycoproteins and glycolipids. So from the endoplasmic reticulum, there could come either the protein or there could come the lipid. So whether a lipid comes or whether a protein comes, the major function of the Golgi is glycosylation, that is adding of sugar molecules to the protein or the lipid and further formation of glycoprotein and glycolipid. So again, this was an important paragraph and I hope that you all understood it. Now let's move to lysosome, which is another membrane bound organelle. However, lysosomes play a different role, right? So these are membrane bound vesicular structures formed by the process of packaging in the Golgi apparatus. The isolated lysosome vesicles have been found to be very rich in almost all types of hydrolytic enzymes, optimally active at the acidic pH. So this is an important line. Lysosomes would only be active at an acidic pH. If you give it a basic pH, the lysosome will not function. Alright, so these enzymes are capable of digesting carbohydrates, proteins, lipids, and nucleic acids. Let's move to vacuoles. What are vacuoles? What roles do they play? So the vacuole is the membrane bound space. Again, vacuole is again membrane bound, right? So because these four are a part of the endomembrane system, which consists of ER, Golgi, vacuole, and lysosomes, right? So vacuoles are membrane bound space found in cytoplasm. It contains water, sap, excretory products, and other materials which are not useful for the cell. The vacuole is bound by a single membrane called tonoplast. In membrane cells, vacuoles can occupy up to 90% of the volume of the cell. In plants, the tonoplast facilitates the transport of a number of ions and other materials against concentration gradients into the vacuole. Hence, their concentration is significantly higher in the vacuole than in the cytoplasm. Okay, so the concentration of ions is significantly higher inside the vacuoles. In amoeba, the contractile vacuole is important for excretion. In many cells, as protists, food vacuoles are formed. So the role of contractile vacuole is what? Excretion. So basically, you can say the vacuole is sort of like the dustbin of the cell. It contains a lot of things that are not needed by the cell anymore. Let's move to mitochondria, which is a double membrane structure, like your ER and Golgi, double membrane. So mitochondria, unless specifically stained, are not easily visible under the microscope. So to visualize mitochondria, usually we use electron microscopy. The number of mitochondria per cell is variable depending on the physiological activity of the cell. For example, if a cell is present, let's say, in a location where more energy is needed, for example, your muscle cells have higher amount of mitochondria, and your neurons also have a lot of mitochondria, right? So because these cells are actively doing work, and to do work a lot of energy is needed, and mitochondria is, as you all know, the powerhouse of the cell, right? In terms of shape and size, also considerable degree of variability is observed. Typically, it is sausage shaped or cylindrical, having a diameter of blah blah blah. Each mitochondria is a double membrane bound structure with the outer membrane and inner membrane. So from this paragraph, only the fact that it is a double membrane structure is important from NEET point of view. The other details like the size, I don't think it will come, but if you want, you can make a chart of the sizes of different organelles for your own curiosity. All right, let's move ahead. So the membrane dividing the lumen distinctly into two aqueous compartments, the outer compartment and the inner compartment. The inner compartment is called the matrix. Now this is the important part. So mitochondria is divided into compartments. The inner compartment called the matrix. The outer compartment forms continuous limiting boundary of the organelle. The inner membrane forms a number of enfoldings called the cristae. So you know how it actually looks. It's somewhat like, it looks like this. So these things are known as cristae, and this is the matrix. Okay. So the outer membrane forms towards the middle. The cristae increases the surface area. The major role of cristae is to increase the surface area. Two membrane have their own specific enzymes associated with the mitochondrial function. Mitochondria are the size for aerobic respiration. They produce cellular energy in the form of ATP. Hence, they are called powerhouse of the cell. The matrix also possesses a single circular DNA molecule. Now, questions have come from this part that chloroplast and mitochondria, what is the do they have genetic material? Then yes, both chloroplast as well as mitochondria, which are both the part of the endosymbiont theory, have their own ribosomes. They have their own genetic material, and they are capable of producing their own proteins as well. Now the important thing is that the genetic material that is present inside the mitochondria is circular and double-stranded. So mark it, this question has come previously and chances are it might come again, because see, the in a human cell, there is a nucleus. There is also mitochondria. So mitochondria also has DNA, nucleus also has DNA. Now the DNA that is present inside the nucleus is linear and double-stranded. And the genetic material in the mitochondria is circular and double-stranded. All right. So remember this. Few RNA molecules, ribosomes, and the components required for synthesis of proteins, the mitochondria divide by fission. This whole paragraph is important from the point of view of your NEET UG. The ribosomes that are present, again, if this is an animal cell, the ribosomes that are present in the animal cell, that for example humans, is 80S, but the ribosomes that are present inside the mitochondria are 70S. Okay. Now let's talk about plastids which are present inside plants. Plastids are found in all plant cells and in euglenoids. They are easily observed under the microscope as they are large. They bear some specific pigments that's imparting specific colors to the plants. Based on the type of pigments, plastids can be classified into chloroplast, chromoplast, and leucoplast. Okay. Chloroplasts have green pigment, chromoplasts have different colored pigment. The function of leucoplasts is to store food. Okay. So the chloroplast contains chlorophyll and carotenoid pigments which are responsible for trapping light energy. In chromoplast, fat-soluble carotenoid pigments like carotenes, xanthophylls and other are present which gives it a distinct color. This gives the part of the plant a yellow, orange or red color. The leucoplasts are the colorless plastids. Okay, so leucoplasts are colorless plastids of varied shapes and sizes which store nutrients. For example, okay, just let me remove this here. For example, a myeloplast which store carbohydrates, that is starch, and elioplast which stores oils and fats, whereas a uroplast stores proteins. So till now questions have not come from this part, but chances are that it might come because it is an important line. It's a very important line. So remember this. The majority of the chloroplasts of the green plants are formed in the mesophyll cells of the leaves, mesophyll cells. These are lens-shaped, oral, spherical, discoid, or even ribbon-like organelles having variable length and width. Their number varies from 1 per cell of the Chlamydomonas, a green algae, per cell to the mesophyll. Like mitochondria, the chloroplasts are also double membrane. Yes. Now this is an important line, that mitochondria as well as chloroplasts are double membraned organelles. Of the two, the inner chloroplast membrane is relatively less permeable. The space limited by the inner membrane is called the stroma. Let me just quickly remove this drawing so I can draw the mitochondria as well. So the mitochondria, for example, looks like this. 4, 5 joint. 1, 2, 3, 4, 5. Again joint 1, 2, 3. So and since it is double membrane, so let me draw the second membrane as well. Yeah. So a number of organized flattened membranous sacs called thylakoids. These are called as thylakoids are present in the stroma. Thylakoids are arranged in stacks like the piles of coins called grana. Okay, thylakoids are arranged in stacks like piles of coins. Over here also you can see thylakoid and grana. Okay. Or the integral thylakoids. In addition, there are flat membranous tubules called the stroma lamellae connecting the thylakoids. This is the stroma. The membrane of the thylakoids includes a space called a lumen. The stroma of the chloroplast contains enzymes required for the synthesis of carbohydrates and proteins. So even chloroplast can make its own proteins, right? It also contains small double-stranded circular DNA. Again, double-stranded circular DNA and ribosomes. Chlorophyll pigments, this is important. Chlorophyll pigments are present in thylakoids. The ribosomes of the chloroplast are smaller, 70s. Again, for example, like your mitochondria was 70s. Similarly, your chloroplast is also 70s. Okay. The cytoplasmic, then the cytoplasm. Then let's just talk about the ribosomes. So ribosomes are granular structures, dense particles by George Pollard, composed of ribonucleic acid and proteins. So ribosomes are basically composed of RNA and proteins, right? And around, for example, in eukaryotic cells, around 80 proteins and your RNA together make up your ribosome. The eukaryotic ribosomes are 80s while the prokaryotic are 70s. Here S represents the Swedberg's unit, indirect measure of density and size. So these are not direct measures. For example, when we were talking about the prokaryotes, we said that 50s bigger subunit, 30s smaller subunit. But when come together, they form 70s. Technically, it should be 50 plus 30, that is 80. But since they are an indirect measurement of density and size and not a direct measurement, therefore therefore this variation in your calculation comes up. So the ribosomes of both uh, prokaryotes and eukaryotes are composed of two subunits. In case of prokaryotes, it's 50s and 30s, and in case of eukaryotes, it's 60s and 40s. So let's move ahead to cytoskeleton. So what is exactly cytoskeleton? Questions have come from this part, so let's be attentive. So an elaborate network of filamentous proteinaceous structures present in the cytoplasm is collectively referred to as the cytoskeleton. So what is it? It's a proteinaceous structure, filamentous and proteinaceous. The cytoskeleton in a cell are involved in many functions such as mechanical support, motility, maintenance of the shape and shape of the cells. So mechanical support, motility, etc. But I mean, this is for your own knowledge. You should know that the function, it's like pillars, right? So like pillars, or you can say both the pillars, the stairs, the lifts, that is the cytoskeleton. So basically the pillars are supporting the whole structure of the building. So that is what the cytoskeleton does. Also the stairs, which are not only supporting but also connecting different floors, right? So they give mechanical support, they give motility, for example, lifts and stairs, and helps in the maintenance of the shape. Okay, now let's talk about the cilia and flagella. So cilia and flagella, cilia and flagella are hair-like outgrowths of the cell membrane. Cilia are small structures which work like oars, causing the movement. So the major role of cilia is in movement of either the cell or the surrounding fluid. Flagella are comparatively longer and responsible for cell movement. Both of them are responsible for movement, right? Uh, The prokaryotic bacteria also possesses flagella, but these are structurally different from that of eukaryotic flagella. The electron microscope study of cilium or flagellum show that they are covered with plasma membrane. Okay, so both the cilia and flagella are covered with plasma membrane. If this is the cell and this is the flagella, for example, it is also covered with plasma membrane. Okay, their core is called the axoneme. So and possesses a number of microtubules running parallel to the long axis. For example, over here, section of cilia and flagella showing different parts. So over here, you can see radial spoke, central sheath, tubular macrotubules, interdoublet bridge. Now, this is an important figure. This figure might come in your exams, in your schools, as well as in your board exams. Their core called the axoneme possesses a number of microtubules running parallel to the long axis. So from this line, you just have to remember that axoneme is present in cilia and flagella. Okay, that's it. The axoneme usually has 9 pairs of doublets of radially arranged peripheral microtubule and a pair of centrally located microtubules. Such an arrangement of axonemal microtubules is referred to as 9 plus 2 array. You can remember this line as well. But don't get too focused on remembering all the details of this line because you may get confused. I mean, I used to get confused, so that's why I'm saying that just remember the parts that need to be remembered. You don't have to remember the whole NCRT. All right. So the central tubules are connected by bridges and is also enclosed by a central sheath, which is connected to one of the tubules. Thus, there are 9 radial spokes total. This is something that you have to remember. There are 9 radial spokes. The peripheral repressors are also interconnected by linkers. Both the cilia and flagella emerge from centriole-like structures called basal bodies. So you can remember this as well, that they both emerge from centriole-like organelles called basal bodies. Now, what is centrosome and centrioles? So we have been talking about centrioles quite a lot. So centrioles are basically membraneless. They are found only in the animal cells. So what are centrioles? They are also responsible for the formation of your cilia and flagella. So they are pretty important, right? So centrosome is an organelle usually containing two cylindrical structures called centrioles. They are surrounded by materials, both the centrioles and the centrosome. Okay, so this part you have to read for your own understanding. Questions have not really come from this part except for the past parts that I have already told you about. Let's move ahead. So, each of the peripheral fibril is a triplet. The adjacent triplets are also linked. The central part of the proximal region of the centriole is a proteinaceous, is also proteinaceous and called the hub. This you can remember. Central part of the proximal region of centriole is proteinaceous and called hub, which is connected with tubules and the peripheral triplets by radial spokes. Okay, no, you don't have to remember this part, at least not for NEET UG. The centrioles from the basal body of cilia, this is important. The centrioles from the basal body of cilia and flagella have spindle fibers that give rise to spindle apparatus during cell division. This line is important. Now let's come to our major hero, that is the nucleus. So nucleus as a cell organelle was first described by Brown. Later, the chromatin of the nucleus stained by basic dye is given the name chromatin by Fleming. The interface nucleus has highly extended and elaborate nucleoprotein fibers called chromatin, nucleoli, perinuclear. Okay, nucleoli. Electron microscopy has revealed that the nuclear envelope, which consists of two parallel membranes with a space between called perinuclear space, forms a barrier between the materials present inside the nucleus and that of the cytoplasm. The outer membrane usually remains continuous with the endoplasmic reticulum and also bears ribosomes on it. Okay, from this, I would give you a detail. So actually what happens is that if this is your nucleus, this is your nucleus, right? So the nucleus is also a double membrane bound structure. From the outer membrane of the nucleus extends the endoplasmic reticulum. Right? And hence the ribosomes which are present. Then whenever there is supposed to be an entry of any protein from outside to outside the nucleus to inside the nucleus, they have to cross this area, right? So inside it is present the nucleoplasm. This is the nucleoplasm. This is the nuclear pore, and this is the nuclear membrane. Inside the nucleus is present a dense part known as the nucleolus. Inside the nucleolus is the synthesis of rRNA, that is ribosomal RNA synthesis. Over here in the nucleus is present your dense linear DNA, right? And different sort of nuclear proteins are also present, nuclear proteins. From this DNA arises your mRNA, that is messenger RNA. This messenger RNA eventually goes out used from the nuclear pore and is taken to the ribosome. From this part emerges your proteins. These proteins then eventually move and go out to your Golgi, right? So this is the whole flow. Now inside the nucleus is present the genetic material. You may recall that at the interphase nucleus have a loose and indistinct network of nucleoprotein fibers called chromatin. But during different stages of cell division, cells show structured chromosomes in place of the nucleus. Chromosomes contains DNA. So basically your nucleus has your chromosomes. Chromosomes are basically composed of chromatin, right? So chromatin contains your DNA plus histones. So chromatin contains DNA and some basic proteins called histones. Some non-histone proteins also RNA. A single human cell has approximately 2-meter long thread of DNA distributed among its 46 chromosomes. You will study the details, blah blah blah. Every chromosome essentially has a primary constriction or the centromere. So this part centromere. This part is known as a centromere. This part is known as a centromere. So what happens if this is your chromosome? The primary constriction is known as centromere. This centromere is eventually enveloped. It is enveloped by a proteinaceous ring known as kinetochore. Now this kinetochore is responsible for attachment of the whole structure to spindle fibers during the process of replication. Alright, so we'll discuss more about it when we'll be talking about cell division. But till now, let's just focus on this, that every chromosome essentially has a primary constriction of the centromere on the sites of which the disc-shaped structure called kinetochore are present. Based on the position of the chromosome can be classified into 4 types. Metacentric. The chromosomes have middle centromere. So in metacentric, I have the structure on the next slide, so I'll be talking about it in the next slide. But for now, the metacentric chromosome has middle centromere forming 2 equal arms. So when we are talking about equal arms, we are talking about metacentric. This part is important. Questions have come, so focus over here. Metacentric, 2 equal arms. Submetacentric, slightly away from the middle, resulting into 1 shorter arm and 1 longer arm. In case of acrocentric chromosomes, the centromere is situated close to its end. Close to its end, not at the end. Okay. So there's a difference between the two statements. When you're saying close to the end, you will mark acrocentric. When you're talking about at the end, you will say it is telocentric. Okay. So in case of acrocentric, the centriole is situated close to its end, forming one extremely short and one very long arm, whereas the telocentric chromosome has a terminal centromere. When we are talking about terminal or at the end, then we will say telocentric. When we are saying near to the end, it will be acrocentric. Okay. So sometimes a few chromosomes have non-staining secondary constrictions at constant localization. This gives the appearance of fragment known as satellite, which we saw in the last slide. Now finally, microbodies. So what are microbodies? They are membrane-bound minute vesicles that contain various enzymes and are present in both plant and animal cells. Okay, questions have not really come from macrobodies, but you can read about it. It's important in that sense. And as I said, metacentrics, that means equal. Submetacentric, this is short arm, this is long arm. Acrocentric, towards the end, but not at the end. And then finally, telocentric, that is towards the end. Okay. So till now, we have been discussing about the chapter. Let us now dive into the last 10 years of questions that have come from this chapter that are very carefully curated so that you can also get a clear idea of what sort of questions are asked. So let's move ahead. So the first question that we can see is we have to match. So whenever these sort of questions come, what I generally prefer is to use deductions. Like if I do not know the answer of all 4 of them, and even if I know the answer of at least 2 of them, I can, mark accordingly. So over here, nucleolus, I know that it is, I've already discussed it with you also. So if you have been noting down whatever I've been saying, and if you have been serious during the class, you will realize that nucleolus is a site of active ribosomal RNA synthesis. Centriole is organization like cartwheel, right? Sorry. The way I drew it was wrong. So centriole is this. Leucoplast is for storing nutrients. Golgi apparatus is the site for formation of glycolipid. So D1, 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 we have 2D1 and then A3. We have 2A3. Then leucoplast C4, C4. This is our answer. Let's move ahead. DNA present in chloroplast, I have already discussed it during the class that the DNA present in chloroplast is circular and double-stranded. So this one should have been easy. Moving ahead with another matching. So satellite DNA, as we know, it was present in the chromosome. Satellite was present in chromosome. Crista is present in mitochondria. Cartwheel pattern is present in cilia and flagella, axoneme and centriole. Okay. Axoneme is present in cilia and flagella actually, and cartwheel pattern is present in centriole. So D3, C4, C4, D3. This is our answer. Moving ahead, mitochondria and chloroplast. Given below are two statements, mitochondria and chloroplast both have double membrane bound organelles. Both are double membrane bound organelles. This is true. Inner membrane of mitochondria is relatively less permeable as compared to chloroplast. So there is no clear indication of a statement like this. So we will say that statement 1 is correct, but statement 2 is incorrect. Moving ahead, in bacteria, the mesosomes are formed by the extensions of plasma membrane. This is true. In mesosomes, the mesosome in bacteria help in DNA replication and cell wall formation. There is no indication of anything like that. So statement 1 is correct and statement 2 is incorrect. Moving ahead, which of the following statements are correct with respect to Golgi apparatus? Golgi apparatus is an important site of formation of glycopore. This is true. It produces cellular energy. Golgi does not produce ATP. Mitochondria produces ATP. It modifies the protein synthesized by ribosome on ER. This is true. It facilitates the transport of ions. It provides mechanical support. So A and C. This is the correct one. Transport of ions is facilitated by channels and transporters that are present on the plasma membrane. It's mostly proteins that do this job. And as for the mechanical support, cytoskeleton is responsible for mechanical support, and ATP is mitochondria. So you have to be very careful when you're reading the question so that you do not make any silly mistakes. So in tissue culture, leaf mesophyll cells are put in culture medium to form callus. Okay, this is something that was not present in the chapter, but I am going to discuss it because a question has come up which requires this particular knowledge. So whenever you do tissue culture, so what you basically do, you take, for example, you take a leaf from a plant. The leaf has cells, so you cut a small part of the leaf and you culture it on a Petri plate which has agar along with other nutrients and hormones. From this particular leaf part, you will get a callus. Okay, so callus is sort of undifferentiated mass of cells. Like you can change the ratio of the hormones to make this callus either form a root or a shoot. So it can form anything at this stage. Okay, so callus has no predefined destination. You can give it conditions to make something out of it. Okay. Over here, the leaf has already been differentiated. When we say differentiated, it means that the function of the cells present here is already known, right? So we already know that the cells that are present in the leaf would be responsible for photosynthesis. So it is differentiated. Its role is known. When you cut this leaf and put it on the plate, these are still the cells of the leaf. But eventually when it forms callus, now the differentiation is reversed. So we say it as de-differentiated. Okay. So leaf mesophyll cells are put in culture medium to form callus. This phenomena may be known as de-differentiation. Alright. Now for example, if you give this callus a certain treatment in which the hormone auxin is more or the hormone cytokinin is more, depending upon what conditions you give, it may form either a root or a shoot, right? So if it forms any of these two, then this is known as redifferentiation. That means that again the fate of the cells have been decided depending upon the hormone treatment that you give it. Now, how many different proteins does the ribosome consist of? So it consists of 80 different proteins. We have already talked about it in the class. Which of the following are not considered as part of endomembrane system? As you know, as we have already talked about, endomembrane system consists of basically four organelles. One is endoplasmic reticulum, one is Golgi, one is vacuole, and the last one is lysosome. So from here, mitochondria is not a part, ER is a part, chloroplast is not a part, Golgi is a part, peroxisome is not a part. So B and D only. Let's move ahead. Which of the following functions is carried out by cytoskeleton? Cytoskeleton, transportation. Hmm. Okay, even I'm confused in this question because motility also seems to be. Uh, okay, so I would go with motility because I gave you that example, no, that it's a building and cytoskeleton helps in plays a role like stairs, right? I gave you that example, and from stairs, I generally correlate motility. So I'm assuming that motility would be the correct answer for this one. In case it is not the correct answer, please mention it in the comments so that I can also know, and others who will be watching the video will also not make the mistake. However, I think motility would be the answer. So the phenomenon by which the undividing parenchyma cells start to divide mitotically during plant tissue culture is known as undividing parenchymal cell. This means that it has been differentiated. That means the function is already known. That's why it has stopped to divide, right? Undividing, that means it has become a permanent cell. The role is decided. Start to divide mitotically during plant tissue culture. So this is known as de-differentiation. Then, okay, leg hemoglobin. This is not exactly from this chapter, but from our prior knowledge, we can try to answer it. Porins are pores. It's as simple as that. Then respiration is an amphibolic pathway in the sense that it is both a catabolic as well as anabolic process. Okay. Then leg hemoglobin, pink colored nodules. So H accumulation in, proton accumulation in lumen. So A4. Okay, from only one option we could have gotten the correct answer. That is what I'm saying, that sometimes when you see questions like these in the exams, do not get all tensed and don't give up. Even if you don't know the rest of the answers, as long as you know even one of them, chances are you might be able to solve the question. Which type of substance would face difficulty in passing through the cell membrane? So as we talked about, cell membrane is composed of bilayer of phospholipids. So anything that is, lipidy would be able to move faster, and anything which is more of watery sort of thing, hydrophilic substances would face difficulty. So substance soluble in lipid would be able to pass very easily. Hydrophobic moiety, that is the same thing. So it would be able to pass easily. Substance with hydrophilic, this will have very much difficulty in passing through oil. It's like water passing through oil. Okay. Then if the pH of lysosome is increased to alkaline, then the lysosome will be non-functional. It will be hydrolytic enzymes will become inactive. Which of the following statements with respect to ER is incorrect? RER has ribosomes. ER is divide of ribosome. Prokaryotes only RER is present. In prokaryotes, this is the incorrect statement because prokaryotes do not have endoplasmic reticulum because they do not have membrane bound organelles, and endoplasmic reticulum is a double membrane bound organelle. Which of the following is an incorrect statement? Nuclear pores act as passages for proteins and RNA molecules in both directions. This is true. Mature sieve cube elements possess a conspicuous nucleus. Macrobodies are present both in plant and animal cells. Okay, so I think this is the incorrect statement. Microbodies are present both in plant and animal cells. Because the first statement is true. Yes, the second statement is also true, and the fourth statement, perinuclear space forms a barrier between the materials present inside the nucleus and that of the cytoplasm. This is also true. So the third statement is the incorrect statement. Moving ahead, Christie are enfoldings in mitochondria. Thylakoids are flattened membranous sacs and stroma. Centromere is primary constriction in the chromosome. Cisternae are Golgi apparatus. So A3B4. A3B4. This is our answer. When the centromere is situated in the middle of two arms, in the middle of two equal arms, so equal arms, it means metacentric. We have already discussed it previously as well. The organelles that are included in endomembrane system. So there were four organelles: Golgi, lysosome, and vacuole. This is our answer. Which of the following statements about inclusion bodies is incorrect? They are involved in ingestion of food particles. They lie free in the cytoplasm. They represent reserve material. They are not bound by any membrane. So this is the incorrect statement, because they are not responsible for ingestion of food particles. Okay, don't make this mistake. It looks like it should be responsible, but it is not. Moving ahead, what is the important site for formation of glycoproteins? Golgi. Golgi and glycosylation always go together. Which of the following pair of organelles does not contain DNA? So mitochondria contains DNA, so this cannot be our answer. Mitochondria cannot be our answer. Chloroplast cannot be our answer, because mitochondria and chloroplast have DNA. So our correct answer would be lysosome and vacuoles. Which of the following statements regarding mitochondria is incorrect? Inner membrane is convoluted. Mitochondrial matrix contains a single circular DNA molecule and ribosomes. Mitochondrial matrix contains a and ribosomes. Outer membrane is permeable. Enzyme are embedded. Mm, Incorrect. Inner membrane is convoluted. So this is the mitochondria, and its inner membranes are convoluted, so that statement is true. This is true. This is true. Mitochondria matrix contains single circular DNA molecule. This is also true. Circular DNA molecule. Yeah. Permeable to monomers. Okay, this statement is the incorrect statement. This is false. So this will be our answer. Enzymes of electron transport chain are okay. No, sorry, I was wrong. The enzymes of the electron transport chain are embedded in the inner membrane, right? If you remember the chapter on respiration, you will realize that the enzymes are present here, here, and not on the, why would they be present in the outer surface? Because the enzymes need to act inside the mitochondria, right? So they cannot be present on the outer surface, because if they are present in the outer membrane, then they will not be able to function, because the enzymes need to function inside the mitochondria for the mitochondria to be able to do aerobic respiration, right? So our correct answer would be option D. So which of the following statement is not correct? Lysosomes are membrane bound structures. Yes, lysosome has a single membrane. Lysosomes are formed by process of packaging in the ER. Lysosomes have numerous hydrolytic enzymes. This is also true. Are active under acidic pH. This is true. This is true, and this is true. So this is the wrong statement. Okay. Because they are not formed by the process of packaging in ER. They are formed by the process of packaging at Golgi. Packaging occurs at Golgi. Okay. The Golgi complex participates in formation of secretory vesicles. Activation of amino acids does not happen in Golgi at all. Fatty acid breakdown is also not the role of Golgi. Respiration is obviously the role of mitochondria. So yeah. Which of the following is true for nucleolus? It takes part in spindle formation. Not at all true. Larger nucleoli are present in the membrane-bound structure. It is a site for. This is the correct statement, and I have already discussed it in the class as well. Which of the following events does not occur in RER? RER. So what happens in RER is that this is, for example, the ribosome. This is the mitochondria. From it comes out a folded protein. This protein has, for example, this is the signal peptide. This signal peptide causes this protein to enter inside the ER. When it comes inside the ER, this signal is cleaved. So this happens. Then phospholipid synthesis. This happens in smooth endoplasmic reticulum. Then protein folding and protein glycosylation. So Protein folding actually occurs before it enters, and but protein glycosylation also occurs in Golgi. So both of these are can be the answers, but I would go with this because protein glycosylation is a function of Golgi apparatus. Okay. Select the incorrect match. Allosomes, X chromosomes. Yes. Autosomes are your normal chromosomes and allosomes are your sex chromosomes. Submetacentric, submetacentric. Lambush chromosomes are diploidine bivalence, polytene chromosomes. Okay, so when we will be talking about cell division, I will talk about these topics as such, but over here this statement is correct, this statement is also correct. Lamprecht chromosomes we see in case of diploidine bivalence. In case of polytene chromosomes, we see it in uh, Drosophila. All right. So, which of the following cell organisms is responsible for extracting energy from carbohydrates? Mitochondria, this is a simple one, everyone knows this. Select the wrong statement. Pili and Fimbri are mainly involved in motility. This is the wrong statement. We have already discussed this, right? Pili is involved in bacterial reproduction. Yeah, bacterial reproduction. Fimbri is involved in attachment. They're not involved in motility. So the very first option was a wrong statement. So a cell organelle containing hydrolytic enzymes, this is easy, this is lysosomes. Select the mismatch, large central vacuole animal cells. This is the mismatch because large central vacuole is present in the plant cells. Moving ahead, mitochondria and chloroplast are semi-autonomous organelles. Yes, because semi-autonomous organelles are those organelles which have their own DNA and which have their own ribosomes. uh, and both the chloroplast and mitochondria meet these conditions. So both of these organelles are semi-autonomous. They're formed by division of pre-existing organelles and they contain DNA but lack protein synthesis. So this is a false statement because they do have a protein synthesizing machinery because they do have ribosomes. So statement A is true but B is false. Moving ahead, which of the following cell organelles is enclosed by a single membrane? So mitochondria is double membrane, chloroplast is double membrane, lysosome is single membrane. So this is our answer. Nuclei again is a double membrane. Now we have to match the columns. So cisternae, Christie is in foldings in mitochondria. Cisternae is let's go step by step. My mind is sort of fogged. I've been talking for the past 1 hour, 35 minutes or something. So, chromatin is the condensed structure of DNA. Cisternae is disc-shaped sacs and Golgi. Thylakoids are flat membranous sacs and stroma of chloroplast. Alright. Which of the following structures is not found in a prokaryotic cell? So, of course, nuclear envelope, because in the prokaryotic cell, the genetic material is freely lying around and it is not enveloped by anything, like in case of eukaryotes. Then cellular organelles with membranes are ER. Ribosome does not have a membrane. Lysosome, Golgi, and so this will be our answer. Whenever you see ribosome, those cannot be the answer because ribosome does not have a membrane. Which of the following structures is not found in prokaryotic cells? So prokaryotic nuclear envelope, as we have already discussed. Then which of the following are not membrane bound ribosomes? See, I told you while discussing this topic, right? There's a lot of questions that come from this topic. So it is one of the most important topics from this chapter. DNA is not present in ribosomes because nucleus has linear double-stranded, chloroplast has circular double-stranded, and mitochondria also has circular double-stranded DNA. Okay, so nuclear envelope is a derivative of rough endoplasmic reticulum. because during replication the nuclear envelope first disappears, and then again when the division is complete, from the smooth endoplasmic reticulum, uh, from the rough endoplasmic reticulum, it comes and forms again. So it is a derivative of rough endoplasmic reticulum. The structures that are formed by stacking of or organized stratum membranous sacs in the chloroplast are known as grana. The chromosomes in which centromere is situated close to one end. So if it would have said that when the centromere is at the end, I would have gone with telocentric, but since it is saying situated close to one end, I would go with acrocentric. Then, which one of the following is not an inclusion body found in prokaryotes? Synoviation granule, phosphate granule, polysome, glycogen granule. So, polysome is obviously not an inclusion body, because what is polysome? Polysome is when multiple ribosomes attach to a single mRNA. This is known as a polysome. So, it is not an inclusion body. So the last question of the session is that select the correct matching. So over here, from the options only, as soon as so my eyesight first read on the last option, which is smoothia, that's a synthesis of glycolipids. This is the correct function. Raffia synthesis of glycogen. This is wrong. Raffia is basically responsible for some modifications on the protein. Smoothie R, oxidation of phospholipids is wrong. It is for formation of lipids. Rafi R, oxidation of fatty acids is wrong. It's for formation of proteins. So that's it from my side. If you all have any more questions and anything that you would want to discuss, please reach out to me. I hope this session was useful for you. And I don't know, this is my first video, so it's I have to ask for this, right? Please like, share, and subscribe, and hit the play button to keep yourself updated about any recent videos that I will be uploading. It will be useful for your own NEET preparation, and yeah, I'm always there to help you whenever you need it. Okay, that's it from my side. Okay, bye.